Welcome, dear students, to today's class on adrenal medullary hormones. So, the adrenal medulla releases catecholamines, and the catecholamines are the structure includes two major structures, that is catechol and amine together. So, and the most important of the hormones are the epinephrine, norepinephrine, and dopamine. These hormones are water soluble, and 50% of it is bound as to plasma proteins. There is no specific transport protein, and these hormones are produced by the chromaffin cells of adrenal medulla. Half-life is almost approximately 2 minutes. So in this figure you can see that several factors act on the hypothalamus, medulla and the pons, including hypothermia, hyperglycemia, hypovolemia. Hypovolemia is a condition where your blood volume is very low, trauma, pain, anxiety, etc. So these factors or these external factors can activate the hypothalamus and thereby activating the sympathetic system, which activates the adrenal gland, and there is a release of acetylcholine causing the release of epinephrine, and epinephrine acts on distant target cells. At the same time, acetylcholine also acts on the sympathetic ganglion, causing the release of norepinephrine, which again acts on target cells at the point of release. So the synthesis of catecholamines is from tyrosine. It's a very small pathway, that is, the tyrosine undergoes hydroxylation by tyrosine hydroxylase. There's an extra OH group that is added over here, and the compound is known as DOPA. Alright, and in the next step, what happens is it is a decarboxylation step. The carbon dioxide is removed, and L-DOPA is converted to dopamine. So it becomes an amino compound, and the dopamine is then hydroxylated. A third OH group is added or at this place, and it's called noradrenaline or norepinephrine. And finally, you have a methyltransferase which adds a methyl group to the amino group. So this becomes adrenal or epinephrine. So in the degradation process, we have seen up to here tyrosine converted to DOPA, DOPA converted to dopamine, dopamine converted to norepinephrine, and norepinephrine converted to epinephrine. So for the degradation, catechol O-methyltransferase and monoamine oxidase are the major enzymes that are involved, and the monoamine oxidases convert dopamine to DOPAC and norepinephrine to normetanephrine, and COMT also acts to convert epinephrine to metanephrine. And the DOPAC is finally converted to homovanillic acid, whereas the normetanephrine is converted to vanillyl mandelic acid. Next is the secretion of the catecholamine hormones, and that is how they are secreted from the cell with the help of external stimulation. That is usually through inhibitory transmitters which act on receptors located in the adrenergic chromaffin cells. As we've seen earlier that the chromaffin cells synthesize the catecholamines, and the activation of several compounds or several chemical compounds like P2Y receptors and alpha-2 adrenergic receptors all inhibit the voltage sensitive calcium channel. This is a voltage sensitive calcium channel, VSCC. So all these you have it over here, P2YR, alpha-2 adrenergic receptors and kappa opioid receptors inhibit the voltage sensitive calcium channels via G inhibitory or G protein coupled inhibitory receptors, and consequently causes exocytosis of the catecholamines. The protein kinase C is negatively coupled to VSCC. So you have the protein kinase C over here. So it is negatively coupled to VSCCs in an isoform specific fashion. So this is an adrenergic cell, noradrenergic chromaffin cell. And the adrenergic cells, you have the voltage sensitive calcium channels which are inhibited by several systems which include P2YR, the GI or G protein coupled inhibitory protein or receptor induced pathways. So the P2YR and the G protein coupled receptors are activated by the presence of ATP, and the ATP can be from the neurons, or the ATP can be from the noradrenergic cells, or the ATP can be from the cell itself. So all of these will activate the inhibitory system, and as a result the phospholipase C is also activated, which leads to synthesis of PKC. And the PKC inhibits voltage sensitive calcium channels, and these compounds also inhibit voltage sensitive calcium channels. So the calcium concentration starts to decrease, and as a result there is exocytosis of the cytoplasmic vesicles which contain the hormones. So in this figure you can see that tyrosine enters into the cell, and tyrosine is converted to DOPA by tyrosine hydroxylase. DOPA is converted to dopamine by decarboxylase. Dopamine enters into the secretory granules, and the secretory granules, inside the secretory granules, the dopamine is converted to norepinephrine, and the norepinephrine can then be exocytosed out of the cell, right? And so the norepinephrine that is exocytosed out of the cell acts on the post-junctional receptor, and as a result, and the remaining whatever is taken up into that, other than that, the extra norepinephrine can either diffuse into the blood, can either act on the pre-junctional receptor, can undergo extra neuronal uptake, can be metabolized to be removed from the system, or it can undergo neuronal reuptake. So there are several fates for the norepinephrine that is released, and also the voltage sensitive calcium channels are the reason that activates the process of exocytosis, right? So, and the alpha 2 receptors which are activated through the norepinephrine released from the cell itself, from the neuron itself, will inhibit further exocytosis.